Okay, welcome back, everybody. We're right on schedule. It's 10:15, and we have our next two presenters who are going to be joining three panelists from New Mexico. So let me first start by introducing Margarita Lopez Uribe and Robyn Underwood, who will be presenting together along with our panelists. So Margarita is an assistant professor of entomology at Penn State. She received her BS in biology from Universidad de los Andes in Colombia, her MS in genetics and evolution from Universidad Federal de São Carlos in Brazil, and her PhD in entomology from Cornell. She was a National Science Foundation postdoc researcher at North Carolina State before coming to Penn State. She's an evolutionary ecologist and extension specialist in pollinator health, and her research focuses on understanding how environmental change and human management shapes bee health and the long-term persistence of their populations in agricultural areas. Margarita has three children, Tiago, age 8, Simona and Leonor, I guess those are twins,
Yes.
5, and she loves playing piano and runs in the summer in her free time when she's not out
Yes.
chasing the bumblebees. And also joining us today will be Robyn Underwood. She's an assistant research professor of entomology at Penn State at the College of Agricultural Sciences. She earned her MS degree in applied ecology from the University of Delaware and her PhD in entomology from the University of Manitoba. She's broadly interested in honeybee colony management and its effects on honeybee health. An interesting fact, Robyn's an entomologist and Robyn's husband is an ornithologist. So technically, they study the birds and the bees. We're so excited to have you here today. I just love your smile. I love your background. So welcome and take it over.
Thank you. Thank you, Steve. So I guess good morning, everyone, right? Like it is afternoon already where I am, but I would like to thank Kathy and Steve for the invitation. As I was saying a few minutes ago, COVID has definitely have a positive impact, bringing people from, like all across the country and every part of the world together through these virtual platforms. So it's very exciting to be joining you today. So I listened to Randy's presentation. It was amazing, lots of information. We're gonna switch gears a little bit for this talk. And so we are going to talk about, like this idea of the beekeeping philosophy. It may be something new to some of you. And the goal of this presentation is to get across what a beekeeping philosophy is and how it matters for the management of your colonies in your operation. So I'm going to start by giving you a little bit of background of, the kind of how this concept came to be, starting with this slide, which is included in that publication at the bottom. It was published in the journal Insects in 2019. And basically, this figure summarizes the whole spectrum of management practices that are available to beekeepers for every given problem, right? So for example, varroamites, Randy just spent a whole hour talking about different methods, how to use them, like how they can be good or bad, right? So there are a lot of options available for beekeepers. But when we think about the level of intervention, there's a whole spectrum too, right? And so there are going to be certain practices that are going to be long lasting or that may change fundamentally the colony, while some other practices may be short intervention or low intervention. And with this idea in mind, we wanted to understand if we could identify things that could explain why beekeepers chose certain management practices over others. So Robyn, Robyn has been working on beekeeping for many years. I'm relatively new to beekeeping research, but Robyn, throughout her, career, she has interacted with a lot of beekeepers, and she started noticing, this idea of beekeepers do have kind of different philosophies about how they approach their operations. So just stepping back for a second, thinking generally about what a philosophy is, it is defined as an attitude or a system of thought that acts as a guiding principle for behavior, right? Doesn't necessarily apply to beekeeping. But when we bring that concept to beekeeping, beekeeping philosophy is defined as the general attitude towards the role of the beekeeper in the lives of honeybees. So if we take that concept, we kind of like develop these three concepts of beekeeping philosophy, which are summarized in this slide. So the first philosophy would group beekeepers who will do anything legal to keep bees alive, right? And so basically the ultimate goal of the beekeeping operation is to keep the colonies alive. The second group, which we call, organic beekeeping philosophy, are beekeepers that, of course, will intervene as needed to keep the colonies alive. But then there is also a priority for bee welfare, right? And so Randy kind of like touched on these during his presentation, kind of like caring about, like keeping healthy bees. And that's a balancing act, right? Like you cannot let the bees die of disease, but also if you overtreat or you use really, nasty, chemicals, that may also have an impact on bee welfare. So that would be kind of like that second category. And the third group of beekeepers from a philosophical perspective, we thought would be beekeepers that do kind of like minimal management, because conceptually, the thought is that the bees know what is best for them, right? And so the role of the beekeeper should be kind of minimized. So these were kind of ideas, right? And perhaps, like these ideas sound familiar, right? Like we all come to a lot of beekeeping meetings, and we meet people, who think differently about this and approach beekeeping in different ways. But we didn't really have hard data to show that these beekeeping philosophies do indeed exist. So to solve this problem, what we did is we took the data from the questionnaire that is, or the survey that is collected by the Bee Informed Partnership. If you haven't heard from these group, it is, like they do a very, very important job in the United States. They send these surveys and collect information all across the country. It's a very in-depth survey. I think it's close to 100 questions where, they collect information about practices, survival. It's a very, very detailed questionnaire. And so we purchase the results of that survey for 3 years, and we use that information to analyze the data and see if we could see correlations between what we see, like what we define in this philosophy and the sorts of management practices that beekeepers are using overall. So it may sound a little bit abstract, what I'm saying. So what we're going to do now is I'm going to ask you guys to bring a piece of paper and a pen or a pencil, and we are just going to go over a few questions that we use for our study. And these are questions that are included in the Bee Informed Partnership questionnaire. And what I would like you to do is to write down your answers. I'm just going to go over 4 questions. And then at the end, we're going to tally points that are associated with your answers. And then you're going to get some, like sense or proxy for what the beekeeping philosophy in your operation is. So I'm going to just check the chat to see, responses. Okay. So I guess, I just want to know, folks, can you write, ready, or yes, I have paper, just to make sure that, we're kind of like all set. Because it is important that we all write questions. Okay, so I'm giving some thank you. Awesome. Okay, so here we go. First question is, which, if any, of the following did you apply to any of your colonies last year? So the first option is synthetics, for example, amatraz. The second, with the option B, is organic acids like oxalic acid or formic acid. The option C would be essential oils like thymol, and D would be none. And if you applied several of these, right, write them all down, and then we'll tally at the end. Okay. So if you are ready, just want to, like, again, interact with you in the chat, because I cannot see, like, body language. I don't know when people are going to be done. So, okay, cool. We have a few folks ready. Okay, thank you, guys. Okay, second question is, which, if any, of the following did you feed to any of your colonies last year? Okay, so option A would be any type of carbohydrate. B would be carbs with supplements. C would be honey, and D, none. So again, write down your question, your answers to this question. Okay, let's move to the third one. So this one is about queen replacement. How often did you replace your queens in your colony? Or do you, how often do you replace your queens in your colony? So the first one is frequently, or option A is frequently, option B infrequently, and option C only to correct. Option D would be never replace your queens. Okay. And the last one, is about the goals of the beekeeping operation. So if the goal of your beekeeping operation is to produce bees for sale, you would write option A. Honey production would be option B. Just a hobby would be option C, and D would be other. Okay. Thank you for interacting in the chat. So again, if you have kind of like multiple options, like, write them all down. So now what I would like you to do is to tally your results. So for every A that you wrote down, you add 5 points. For every B, you write 3 points, C 1, and D 0 points. Okay, and I know that we have kind of a tight agenda and schedule, so I'm just going to kind of like move with this. So basically, if you tally your results and you added more than 28 points, most likely your beekeeping philosophy aligns with conventional beekeeping. If the points that you tallied are between 6 and 27 points, it's most likely aligns with organic and IPM, and less than 5 points aligns with chemical-free or natural. And so, of course, the study that we did included a lot more questions, so it was more comprehensive, but this kind of like gives you an idea of how we collected the data for the figures that I'm about to share with you. Okay, so in the next few slides, I am going to show you a multivariate analysis. So basically, we took all of the answers from all the beekeepers in the United States for these 3 years. And what we did is we wanted to know how close the overall beekeeping management practices between beekeepers were. So in this plot, right, like this plot is just like the space of all beekeeping management practices. So each point is represent a beekeeper. And so if 2 points are closer to each other, that means that the management practices of those two beekeepers were more similar than points that are farther apart. And the colors are indicating what like we coded as natural, organic, or conventional beekeepers. So for example, if we think about the management practices for supplemental feeding, what do we see? So we see here, like the spectrum, right? Like, so we see beekeepers who feed honey in no protein, carbohydrates with supplement, and proteins in any carbohydrates. What we see is that beekeepers that align with the natural beekeeping philosophy have a greater tendency to feed honey to their bees. Well, the group that aligns with the organic beekeeping management practice is more likely to give carbohydrates with supplements, right? And in the conventional, the other stream, we have beekeepers who are more likely to give any carbohydrate and provide protein supplements for the colony. So this is just one aspect. if we look at the goals of the beekeeping operation, we see again a very similar result with like the clustering of the beekeepers. So a lot of the folks who are kind of like in this philosophy, natural, chemical free, are beekeepers who do not have an expectation of harvesting honey, and the purpose of the operation is really kind of recreational or for teaching or for research purposes. And here we have, there's significant overlap, right? Like the organic and the conventional beekeepers have, the tendency is to, of course, have a kind of an expectation for an income. There are a lot of beekeepers who have this business for honey production and for bee production. However, something else that we learn from this is that another important driver of the choices of beekeepers for how they deal with problems relates to operation size, right? And so here the colors are not going to indicate organic, natural, and conventional. The colors indicate the size of the operations of the beekeepers, but the placement of the points is exactly the same as in the previous ones. So, so here what we find is that the commercial beekeepers, right, are located here. And the overall management practices of these beekeepers is very unique to them, right? And so they have kind of like very different practices when the operation is above 500 colonies. Here we see that sideliners, are located here, and there is a lot of overlap with the sideliners between kind of the conventional management practices. Sorry, okay, so this is an important thing. So these are the size of the operations, but remember that here in this side of the plot, we had all of the conventional beekeepers, right? So all of that, this side of the plot here is showing you beekeepers that have conventional management practices. So the take-home message here is that among the backyard beekeepers, right, so folks who have less than 50 colonies in their operations, we see the greatest variation in management practices. So we have a lot of beekeepers who are natural or aligned with natural philosophy, a lot of beekeepers who align with organic practices, and some beekeepers who also have kind of conventional management practices. While the commercial and sideliners are beekeepers that are going to be following or have philosophies that align more with conventional and organic management practices. Okay, so the take-home message of analyzing these survey data is that we learned that indeed the beekeepers can be clustered depending on the sort of management practices that they use, and that the two main drivers of these is beekeeping philosophy, so conventional, organic, or natural, as we predicted and hypothesized at the beginning. But then the other important factor is operation size. And so really this beekeeping philosophy is going to be more important for beekeepers who have small operations, while the sideliners and commercial are going to be more aligned with either commercial or organic practices. And so the next step after working on this project, defining these philosophies, was really to do a controlled and well-replicated experiment assessing how is it that these different approaches to beekeeping have an impact on colony health. And this is what Robyn is going to talk about and present some preliminary data on. So I'll be happy to answer questions at the end. Robyn, if you want to, I need to stop this, right? Make you share.
Okay, yeah, thanks a lot. So I have been working in charge of this large field project where we're comparing the three philosophies on actual honeybee colonies. And so we applied for and we received a grant from the USDA to conduct this work. And Luckily, we got it early. We got it in September, and we didn't have to get our bees until April of the following year. So we had some time to really think through how we were going to approach this project. So we ended up finding beekeepers in Pennsylvania, because that's where we are, that represent each of these philosophies. So we found 30 people to invite to come together at Penn State and discuss beekeeping philosophies for a whole entire day. So we started off the day, describing the project and things like that. And then in the afternoon, we had them break into the 3 groups. So each beekeeping philosophy had literally a round table discussion. And intermingled in there, we actually invited all of our apiary inspectors. So they had to, they weren't allowed to all go to the same place. They had to scatter. And so there was a couple at each table because we figured the apiary inspectors have seen all of these philosophies as they're visiting beekeepers in Pennsylvania. So we got this great group of people. They're still with us. We still meet with them regularly. And what they did is they answered a myriad of questions that we had for them and helped us to shape these management systems that we used in practice. So very briefly, you probably can understand that the conventional system is the most inclusive. So in the conventional system, synthetics, organic acid, and cultural controls are all okay. When you switch to organic, though, you eliminate the use of synthetics entirely. And when you go to what we're calling chemical-free here, which you could think of as natural, treatment-free, chemical-free, you can use a cultural control. And what we ended up using was small cell comb, for example, but 0 chemicals are ever put into the colonies for any reason. So I don't want to go through every detail of this, but basically, this is kind of like the beekeeping philosophy continuum where you have to choose, right? So I would encourage you when you, later, if you look up that paper that Margarita talked about in insects with that philosophy, you can download and print that philosophy figure and you can use it as you wish. You can bring it to your club and show people. But if you print it and go through and like circle, I do this, that, you'll be determining your management system. So for our management system, we used all plastic foundation, and that was something that we did not allow our stakeholders to choose because with it being research, we were very strongly interested in comb size. And so we didn't want to, say, give them some sort of foundation to have them draw comb in a different size. So we used standard foundation, except for in chemical-free where they got small cell. Chemical-free also got like a space on the side of each frame where there was an inch for them to build whatever comb they wanted. We also scratched up the inside of the boxes for chemical-free and used a cloth inner cover. And all of that was to encourage the propolis envelope for, self-medication that our stakeholders thought was very important for their health. In terms of treating, in the first year, when they had just started, we only had fall treatments. So conventional got Amitraz and organic got Formic Pro. These 2 got an oxalic acid vapor midwinter. And then in the second year after they were established, and so were the mites, we had to treat more often. And we did that by, just like Randy says, keeping on top of mite numbers. We did alcohol washes every single month. And if they got over a threshold, we treated. So at different times a year, we use different treatments. Because the conventional system is all-inclusive, we were able to use oxalic acid vapor or Formic Pro in the summer, but then come fall, they all got Amitraz. And for organic, Formic acid and Apigard NOA were all things that we could use when the mite levels got too high. These colonies were located in Pennsylvania and also in West Virginia. We had a collaborator do a replicate down there. And so you can see there were 6 different farms in Pennsylvania, 2 farms in West Virginia. Each one of those farms had 36 colonies in 3 apiaries of 12. So it started with 288 colonies. So it was pretty exciting, really big project to get going. But this is what a single apiary would look like. Sadly, we had to build a bear fence for every single one of them. But at the very beginning, we started with 4 of each of those management systems, literally side by side. So just like Randy keeps his, his mite treated and untreated colonies together, we did that as well, because apiary is such a huge influence on your results. So we had to make sure they were all represented there. So they were like in little arcs trying to, improve drifting. We also came in and like put designs on the hives just to try to get them to not go to the other colonies. So this is how we began. We followed we began them with everything, at least inside of those hives, was brand new. So all new foundation. We got new bees in the form of packages. And the reason for that is that for the chemical-free system to work, we really felt that genetics are the key. So we had to find the right genetics that would allow the chemical-free to thrive and then give the same genetics to everyone. And you might have seen on Margarita's figure, there are not beekeepers in the chemical-free or natural system with a lot of colonies. So we couldn't buy 288 of them. They were just not available. So we started with packages from Georgia for chemical-free. We started with normal packages, your standard packages for conventional and organic, but for chemical-free, we got bees from Fat Bee Man because he uses small cell comb, and we wanted to make sure that when they got here, they also built small cell comb. But then come July, we were able to get hundreds of queens from an all-natural beekeeper, and he was able to make sister queens for all of our colonies. So at that point in the first year, we made all the genetics the same. We wanted to be sure that anything that we see is about management and not having anything to do with the genetics of that particular colony. So like I said, we did varroa mite washes every month. We also seasonally collected bees into dry ice for lab testing, which so far we're mostly focusing on fall because we're using it as a predictor of overwintering survival. We of course look at honey production because beekeepers want to have honey. And by the way, I should note that the management systems and how we operated was with full-size stationary honey producing colonies. Our goal was to have an economically beneficial system where you make lots of honey, you might have spare bees and have a good result for people whose goal is to do that. So keep that in mind. That was our whole sort of mode. Of course, overwintering success is critical, and so it was measured every year. We set out to do 2 full years, so you would get from spring to spring 2 times, but we decided to do a third. So these colonies are now this winter overwintering for the 3rd time. So we'll see what that brings. So these are the results of just putting all those colonies onto the same graph with varroa mite mean abundance, using those alcohol washes every month. And you can see a trend here where the mites on the adults stay low throughout the season because, they're reproducing and in the cells, so they're not really seen in the alcohol washes. Then come August, that is when we put our treatments on. Except for not in chemical free, right? The red is totally untreated. So with treating, we were able to keep our mite levels very low. See, this is 2%. So this was a threshold where there's 6 mites in your wash. So they were quite low going into winter. Then of course you can't do varroa washes when it's, winter. So we start again in April, measuring every month. Same thing, we were pretty well able to keep mites under control. Then they started to increase, we treated, or we didn't. Then come October, this is what we think of as, what they're going to into winter with. So then in our 3rd summer, the mite loads were a bit higher. And this past winter was quite mild here in Pennsylvania. So it does seem that perhaps they didn't have a brood break over the winter, or if they did, it was very short. And so we had a bit of more mites in 2020 summer, to the point where our treatments in the fall, so we always do these mid-August, were not effective. So right here, we used Formic Pro for organic and Amitraz for the conventional, and the mite levels were above the threshold that we like in October. So we decided then to do more treating and do oxalic acid vapor 3 times, hoping that the mite levels got low enough for winter. We don't know though, because we didn't do an alcohol wash afterwards. But this was quite interesting and has led me to apply for a grant to see, why would formic acid be ineffective when it's always been effective in the past? And one of my ideas is that there's an impact of humidity that isn't on the label, and no one pays attention to the humidity because they're so focused on the temperature range. So we'll see. Also, Amitraz didn't really work very well either. The mites were still too high. And so I set out and sampled all my colonies using an Amitraz resistance test from Jeff Pettis. And it turns out that there wasn't any resistance. So I can't really explain why the Amitraz did not decrease mite levels. Anyway, every year is different. So it's interesting, always interesting. In terms of honey production, each year organic had significantly higher honey production, definitely higher than conventional here. This is quite dramatic in 2020. Both of them had mites under control. We have to think pretty hard about what potentially could make organic more productive. But this is how everything turned out. Winter survival was quite dramatically different. So what you can see here, this is their very first winter after being in packages and building up for 1 year. We had really good survival in the 2 treatments that we're treating for mites. So organic and conventional, over 80% survival. And if you look around Pennsylvania for that same winter, the average was 50 to 60% survival. So we were pretty happy with our system and moving forward with plenty of colonies. Unfortunately, untreated colonies, even though their mite levels were not above 4% or 4 mites per 100 bees, they still had huge losses. So we only had 36% of our chemical-free colonies surviving that first winter. Then in the second winter, pretty well the same. Organic and conventional both had really good survival, this time over 90%, and chemical-free had 50%. So this was, if you look at the actual number of colonies over time, the chemical-free is down now at this point in time, down to, from the original 96, there's about 6 of them that remain today. So if you try to look at what is causing that, I skipped a few detail slides for time, but what we're looking at here are the lab tests. So here we have vitellogenin. This is sort of a marker of nutrition and health. The varroa mite washes, which management system they were in, levels of deformed wing virus, and this immune gene, defensin-1. So what you can see here in the very first year, the important things that were showing, sort of predicting survival are to the right of this dotted line, positive and negative. So if the colony had higher vitellogenin levels, were in the management system that controlled mites, they were more likely to survive. So this was as predicted. Varroa mites were negatively correlated with, and varroa mites and their associated viruses negatively correlated with survival, as well as this defensive-1 immune gene. So this is sort of what we would have expected and what's generally in the literature. And then we followed them into year 2, and we saw something a little bit different. Defensin-1 still definitely is a predictor. More defensin-1 expression is negative for survival. Vitellogenin as well, and treatment system, the same thing. They're positively correlated with survival. But varroa mites and deformed wing virus were not really important either way in the second year. So we're really, really curious to see what happens in the third year. So this is like a really long longitudinal study following individual colonies for 3 years. So we'll see what happens. So just to sort of summarize everything that we found so far, mites that were in a treatment system, colonies in treatment systems where you control mites had higher survival, lower mite levels. And what that tells me is that the organic system, which by the way made more honey, had excellent survival, and had mites under control, is one that I would go to. It's has a lot of positives, and you don't need to use synthetics. So I think a lot of beekeepers think that they can't get that, synthetic out of their rotation. I believe that you definitely can. In terms of survival, remember management system was really important for survival. So managing mites and disease pressure was sort of important one year and not important another. So this is kind of super interesting to see and try to figure out. So I just want to end this portion of the talk by thanking all the many, many people that have helped us throughout this long process. Managing 288 colonies, many of which are a 2-hour drive to even just get there, took an army, So we thank all the participating beekeepers and all of the people that in the lab and the field, and no one ever does research alone. They always have help. Okay, so we're really curious now and excited to talk to our 3 panelists. And so I'm going to stop sharing because it's really just sort of asking them some questions. So I think if you 3 could all, share video, that would be great. We'd love to see your faces. And I'm going to ask a few questions, and then what's going to happen is each panelist in turn is going to answer the question about themselves. So I don't know if you guys decided what order you want to go in. Craig, do you want to go first?
Sure, I can go first.
Okay, actually, why don't we start by you guys introducing yourselves? And the first question is to describe the essence of your management system in just a few sentences. So that'd be a great introduction.
Right, and so I've used synthetics such as Apivar, which is Amitraz. I do prefer to use oxalic acid fogger. I've been experimenting that with the bees and see what is the best treatment. I also like to do brood breaks. I'll do splits and stuff like that and do other integrated pest management for mite control. I'll requeen as needed, and I'll also supplement feed as needed, and want to work more with nutrition and raising my own queens at some point.
Okay, great. John, you going to go next? We'd like to see you while you're talking, if that's okay.
I'm trying to get my video on. Give me a second here.
Well, maybe Kate, you can go while John's figuring that out.
Sure. So I'm Kate Whalen. I'm up in Santa Fe, run the Beekeepers group up here. I know that we're a little bit colder up here than the Albuquerque area, so some of these things might not apply to Albuquerque.
Okay, thanks.
So I guess my philosophy, I'd consider myself a chemical-free natural beekeeper. I did study with Les Crowder years ago. Many of you are familiar with him. And I use top bar hives exclusively. And I'll kind of paraphrase something that probably he said at some point in time, is that I try and observe the bees, see what they're doing, and I try and help them do it. So I want to play a supportive role. I do make early season splits. I try and keep the colonies small. I don't constantly introduce new bees. I haven't bought any bees since the very first year I started, 15 or 20 years ago. I allow my bees to requeen themselves. I encourage supersedure if that's what they want to do. And I always try and leave the bees enough honey to get them through the spring up until the fruit tree blossoms at least. So that means leaving more honey than most people do. I don't believe in feeding protein. We don't usually have pollen dearth in our area. No chemicals, hard or soft, no plastic in the hives. And my top bar hives I use now predominantly roughs on lumber, which encourages them to propolize. So that's my philosophy.
Okay. John, it's your turn now that you're showing us your feet.
Is my video on?
Yes.
Wonderful, thanks. I got through that process anyway. Thanks for having me on this morning. My name is John Gagne, and we own and manage San Juan apiaries. Our bees are located along the Rio Grande River, south of Santa Fe. Elevation there is about 4,400 feet, I think, so it's significantly different than where my home yard is in Santa Fe. We're at about 7,000 feet. We've been managing bees for a number of years. Actually, I started pre-mite, which kind of gives you an idea of how long I've been in this. We were commercial beekeepers in Virginia, but I've been in the Santa Fe area 31 years. Our goals for our apiaries are honey production. We do use Amatraz. We use Aprovar one-time fall treatment. So I guess if I were to summarize our operation, we're anything but scientific. I've been listening to a lot of scientific philosophy here, and I would love to be able to embrace a lot of those different philosophies in our operation. Essentially, it's just me. So when I get out to the bee yards in the morning during the season, I certainly have a lot of items on the to-do list. Unfortunately, mite washes and a lot of focus on mite management falls by the wayside. Having said that, our yards are very healthy. Actually, I was out looking at some bees last Tuesday. The weather was finally warmed up a little bit. Had a lot of flight activity. Out of 25 hives that I visited in a couple of yards, there was one that looks like didn't make it, just a really small cluster of bees. So we're pretty successful in our management. There's not a lot of negative impact around our bee yards. I don't even know any other beekeepers down where I am. So I think isolation really assists us in keeping our mite counts low.
Okay, so mites are always, the topic of conversation. So my second question is, do you agree with the statement that varroa mites are the biggest threat to beekeeping?
I guess I'll go first since I was the first one, and we'll keep it in the same order, I'm guessing. I don't know. But yes, in part, I also think the nutrition and genetics are key components, as well as beekeepers learning to take care of the bees and adjusting with changes. As John said, I'm sure, and he's adjusted with changes, that beekeeping is different now than what it was when he first started. So.
So Kate here, yes, no, I do agree that, there are some other factors such as nutrition, pesticides in our area, certainly drought and extreme weather events, habitat loss, importing non-locally adapted stock, et cetera. There's a huge list that I think contribute to it, but it does seem like mites or seem to be always listed as the biggest threat. However, and I hesitate to say anything in the presence of an evolutionary ecologist, but there are very well documented groups of bees that have developed varroa resistance in Sweden, in Puerto Rico, in Europe, through careful breeding. So I think that The mites used to somewhat coexist with the bees. If they kill their host, they don't survive either. So there's something out of whack. I do think the bees can overcome this. But at present, if we continue to treat and all the bees are dependent upon treatment, then all of our drum congregation areas are full of stock that needs treatment to survive. And so, you know, when I went to one of the honey producer conventions, even they were saying that if people would just quit treating, let the bees die off, that within 5 years, we would have resistant stock. And obviously that's not going to happen under our conventional agricultural system. I think our agriculture depends upon the bees. I don't think that's an option, but I think on a small scale, we can all continue to strive to deal with this through genetics and heritable traits. So, for what that's worth.
Well, excuse me, I tend to agree with Kate and Craig that in our area, certainly our environment plays a major role in how you have to manage your bees and what the environment, you know, affords. Certainly lack of forage, significant drought in our area for, you know, a number of years. We have good seasons, bad seasons like everyone else, but certainly lack of rainfall, I think, is really critical, which leads to lack of forage, and the bees struggle from time to time, especially if you're a backyard beekeeper or wish to keep your bees in the confines of the city. I mean, springtime in Santa Fe is beautiful with an enormous amount of forage for your bees. After the trees stop blooming, I mean, that's about it. And I know a lot of people struggle in keeping their bees healthy and having enough stores for them. The original question was, are mites probably the main source of bees dying off? And the most important thing, I think, absolutely, is to manage your mites and try to control them the best you can with whatever means works for you. As I mentioned, we're kind of fortunate. We treat once in the fall. I'm not saying that's the best way to do it. For me, as a sole individual trying to manage 80 production colonies and 30, 40 nucleus colonies, my focus is, okay, I'm going to get out there and I'm going to manage the bees the best way I can for that time that I'm out there. So I do not focus a lot on mite washes. We'll sample yards from time to time. But yeah, mites changed the ball game ever since 1989 when I first moved here. And of course, along with that, you've got EFB, AFB, nosema, you know, wax moth control. So it's difficult to keep bees healthy.
Okay, so I know everyone loves their bees and you think that your system is the best one. So if you guys could describe what you think, like to you in beekeeping, what are your definitions of the terms like successful and sustainable?
I guess I need to unmute my mic first. So for me, I think it's a matter, again, what I talked about as far as continuing to learn and adapt to what the bees' needs are. And successful to me means that most of the bees have overwintered more than 1 year. And sustainable means doing splits or nukes to increase the colony size, my colony sizes. This replaces any losses that I have, and it'll help continue improving my queen breeding stock once I get there. I'm hoping to start doing that this year. So.
Robyn, are we supposed to talk about sustainability as well this time, or are we just talking about success? I forget what.
Both success and
Oh, okay.
sustainability.
Okay. So Craig, you might want to talk about if you feel like yours is sustainable, I think was the other question.
Yeah, I think he did. He talked about having colonies that he could split to make up for his losses. So therefore,
Oh, okay.
his operation is sustainable because he can keep the numbers.
Okay.
Right? Is that right, Craig?
Okay. Sorry.
That's
Sorry.
okay.
So, I don't really judge my success by like financially per se. I just sort of like Craig was saying, I feel like if I at least have some colonies that are overwintering, then I can always make up my losses, in the same way that, it sounds like most of the commercial beekeepers have to do. And if my bees are able to produce enough honey for themselves, then I feel like I know that on most years there's enough available forage in my area for the number of colonies that I'm trying to keep in that particular area. But part of, I think, a feeling of success is that I get a lot of pleasure from working with the bees. We have a small farm. They provide pollination. And there's a lot of orchards in our area. And I also do sell honey and a lot of beeswax, which people use for body care products because I use no chemicals. And I think that the accumulation of, it's well documented, that amatraz and any number of chemicals accumulates in wax, and people don't want to use that on their skin. So, but as far as sustainability, I feel like that implies no outside inputs, which to me would include bringing in any chemicals, obviously miticides, protein substitutes, buying outside bees or queens. So I try and avoid all of that. It's not easy, and I'm not saying that this is a hugely financially successful operation, but we're hanging in there. So I feel like it puts new beekeepers at a disadvantage because maybe they don't have the numbers. I think you need a certain number of colonies to be able to accept some losses and then breed from those. And if you only have a few colonies, as a backyard beekeeper, you're always working to try and increase your numbers. And so I think, it's tough, and I can talk a lot about how I've managed it, but I've always been able to, between swarms and cutouts and then just longevity, been able to kind of keep the numbers going. So.
As far as being successful, I think our operation has been very, very successful over the years. We've managed to keep our winter losses very, very low. And we always seem to have more bees than we know what to do with each spring. We do make a lot of splits and make available 5-frame nucleus colonies for sale in the spring. And we always have many more people asking for them than what we have, but again, it's certainly a lot of work. We use it as part of our swarm control management and knock some of these big production colonies down in the spring that they're going to be hanging in trees if you don't do something with them. So we do make a lot of splits. We've been very successful with importing a couple of genetic lines of VSH queens, some from the Baton Rouge labs, some from the Russian Beekeeping Federation. So we've got a good cross section, I think, of genetics in our yards, and of course is always supersedure. We do walkaway splits, which has always been very successful for us. So certain times of the spring, we certainly let the bees do a lot of that work for us. And so, yeah, we're successful. As far as sustainability, I think, again, yeah, we certainly never have had to go elsewhere to look for bees. Year after year, our numbers either stay the same or increase a little bit. We've had to move around a couple of bee yards the last couple of years, so that kind of impact us a little bit. We keep 15 to 20 hives in a yard, so we had to kind of double up on a few of our yards. But we've got some locations this spring. I think we want to start a nuke yard, and just to raise our nuke. So that's looking forward to getting that established. It's interesting to listen to people. I know Kate and I know Craig, and we have different philosophies, but I think the goal is the same. Provide valuable pollination services, although we don't rent bees, but we get a lot of positive feedback from people saying, geez, I'm really glad your bees are there. So I think that's very, very, very important.
Okay, so we're getting close to the end of our time. So if you could each just have like a closing remark, and that could include my last question, which is, what do you think is the future of beekeeping? One sentence each.
This is Craig. I think it looks promising as we again learn to adapt with the environment changes, whether they're good or bad, and then more people as they start working on genetics and learning again and educating people as far as planting crops, planting the pollinator plants, backyard people, even if they don't have bees. I think it looks promising.
Yes, I've been
A lot of
really
sentences.
Oh, how many? Okay.
Go ahead.
All right. I'm just so I'm heartened by the research that has been taking place about VSH traits, studying the grooming behaviors and grooming dances, bees biting off the legs of mites. There is, I think that's where the emphasis needs to be. I just wanted to say also that I'm a big believer in entrance reducers. I think that if people can, people who criticize backyard beekeepers because maybe their colonies are collapsing and causing mite bombs, then you need to really recognize the signs when your bees start crashing, or keeping your small colonies with your entrance reducers on them. And I tend to use a piece of window screen over most of the entrance in my small colonies, so that you prevent robbers and whatever else, mice and wasps. But it's going to reduce the chances that in that period that you're getting used to not treating, that if your mite counts are high, then you're not going to send those mites out to your neighboring beekeepers who might be treating and not want all those extra mites. So anyhow, and the other thing I think might be important is that they're working with almond varieties that are self-fertile. And, I think if they can reduce some of our dependence on commercial beekeeping and all those practices to support agriculture, that'll help our bees in the long term. So.
I think the future of beekeeping looks fine, thanks to our educators, thanks to people like Robyn and Margarita. Your research is extremely important and very, very interesting. Thanks to the Randy Olivers of this world, who know more about mites than most anyone, and he's applied that to his operation, and he's certainly a real trendsetter. So I think it's extremely important from an educational standpoint to have those resources and have people like you who put so much time and effort into it. So I think the future of our beekeeping is fine. Thanks to all the wonderful breeders out there who raise VSH queens and continue to experiment and try to get us a better queen, better genetics. Thank you.
Hey, so I just wanted to break in here and let folks know that we're getting so much positive feedback and so many great questions and comments that we want to continue this. We're gonna give up the break, and we're gonna hope to keep our panelists here. Again, we're getting questions for individual panelists. We're getting questions from Margarita and Robyn. So we'll try to get as many of them in as we can in the next 15 minutes. But this has been a great. We're so glad we invited you here, the panelists, of course, but also our speakers who have really offered us this really brilliant and comprehensive perspective of sort of the span of beekeeping in this country, and the studies that you're doing are really terrific. So let me see if we can pick out a couple of questions to start with. And so, Robyn, you can also help me when I read the question. You might be able to either answer it or direct it to one of our panelists, however you feel best. Let's see, Katie Thompson asks, do you know of any research demonstrating that nutrition can be correlated with mites being less of a pressure in the apiary? If not, it would be interesting to find some apiaries with diverse forage and compare their mite disease rates and productivity with apiaries with less diverse forage. I know diversity of forage has been studied, but has it been directly correlated with mites?
It looks like Margarita has something to say, but in general, I would say that there's a lot of evidence that nutrition matters a lot, right? So I do know that bees would be better able to handle the virus loads if they have high nutrition. I don't know if it would affect mite reproduction or mite numbers, but the associated issues, it's if they have good nutrition, it's one less stressor. So good nutrition is always a good idea.
Great. Let's see. We have some questions also coming in through the comment, through the chat part. So we don't want to miss those as well. Anita, one of our panelists, or previous panelists, is asking, what I've discovered from Dr. Marla Spivak's work on queens and neonics is that it reduces queen fertility and impacts workers' behavior. GPS from pollinating plants with neonics. A comment on pesticides. Does anybody want to talk about that kind of research?
I guess I wanted to have a kind of a follow-up comment to Robyn's response. So well, yes, from the kind of the perspective of the beekeepers, the management of mites is extremely important, right? Like we both Randy in our presentation kind of like shows that. It is not the only factor that is going to drive success or colony survival, right? Like we know that the complex of the problem of honeybee health is extremely complex, right? And so, yes, if you, and we see this in native bees too, right? Like for example, the deforming virus has always been here, right? It's a virus that has been, like in bees for many, many years. But now, right now we have mites that help replicate the virus, low nutrition that suppresses the immune system of the bees. Pesticides also suppress the immune system of the bees. So, if you take the absolute amount of virus and you inject it to a bee that only is dealing with the virus, or inject it to a bee that is having to lead with, poor nutrition and pesticide exposure and, all of other stresses, yes, of course, that is going to increase the probability of, like that bee and the colony dying. So it's always, like we need to keep our minds kind of super open about the complexity of drivers of, colony survival or, death.
Okay, great. We have a comment from Tina in Colorado that talks about her mite numbers climbed high. We treated with various treatments. They didn't work as well as usual. It's very dry in New Mexico and Colorado, so not humidity. she said, this wasn't just me, but across the state in many different microclimates at altitudes from 5,500 to 8,000 feet. I guess it's more of a comment than a question.
Well, I do feel like, treating is complicated. It's not as simple as you throw it on and it always works. So I always encourage everyone to, after the treatment, check again, see if it did work. And if it didn't, you need to respond. Yeah, so I don't know what the explanation would be for that particular anecdote, but certainly everyone should be just being sure that the treatment worked after the fact.
Okay, great. We've had some questions about sort of the spacing of hives and apiaries versus in nature. And so one comment from Christina is that with putting the chemically free hives in the same yard as the conventional, you may have had a mite bomb effect, and maybe it wasn't so much a failure of the treatments, but it had to do with the hives of the different methods being so close to each other.
That's definitely a possibility. The numbers that I saw this fall were so high, I couldn't imagine that like one crashing colony could have made 30 other colonies over the threshold. So to me, the math wasn't adding up, but it's possible there were feral bees in the area. And we do have that data from all of the colonies over the whole length of it. And we can definitely do some analyses to see if, if you're next to or in the same farm as a high mite load colony, are you more likely to have more mites? Like we can do that. We just haven't, we're planning this spring to just dive into the data and look at all of these things.
Well, I think you, along with all the rest of us beekeepers, we typically are ready to dive into whatever we have to dive into. We want to get in there and see how the bees are doing. Matt Ford asked the question, how do you guys attempt to control for genetics during the duration of the study? For example, supersedure, queen loss, etc. Did you replace with similar genetics? Nearly impossible, I know, but just curious.
Yeah, we requeened that one time. And then after that, everything is descended from them and locally mated. So it could have absolutely watered down anything that we got at the beginning, but we sure did our best.
Okay, let's see. There's a question about familiarity with Tom Seeley's work. Let's see, hold on. Have you followed Tom Seeley's work about wild bees when they are able to choose and space their distance between hives and adapt and evolve traits to resist the mites? Any thoughts?
Can I take that one, Robyn? Yeah, so we do actually a lot of work with feral bees in our lab as well. So, one of the reasons why I wanted to answer this question is because we need to keep in mind that resistance to mites and tolerance of mites are two different things, right? And so the outcome of tolerance and resistance is that, colonies can survive at the end of the day. But many colonies achieve that, and this is something that we see in our feral colonies, is through tolerance. They learn how to live with the mites in the colony. It's not that they just get rid of them, right? Like the mites don't go away. They are in the colony, and the colony is just, like they learn how to tolerate viruses, at a higher level, or they swarm more frequently, or the colony size is smaller. So it's just that the dynamics of colonies in feral colonies is very different. And yes, the spacing is extremely important, right? And so what we see is that it would be really, really hard for a beekeeper to maintain, like honeybee colonies at a natural spacing, and Tom Seeley has a lot of research on this, right? Like you could not have an apiary. You would have to have, colonies spaced out, less significantly, and probably not have enough colonies to, like if your goal is to extract honey or have, some economic gains from that. So I guess, like the take-home message, like it is important to separate resistance from tolerance when we're talking about diseases. And sometimes it is hard to really think about honeybees in wild environments and how we can bring, all of that into an apiary and management setting.
Okay, that's great. We have a broad spectrum of attendees as well. And so some folks are really loving the idea of this natural resistance, encouraging natural resistance, survival of the fittest. Others are commenting about sort of our agricultural environment. Just two comments you might be able to respond to. Said maybe mites wouldn't be the problem if we did something about monocropping and poor nutrition. And a follow-on comment with that is we need to talk about habitat that is seriously degraded in the ag sector and even backyard pesticides. Any thoughts about that? Okay, another comment from Amy Owen here in Albuquerque. After 4 years of trying to obtain queens and local genetics that are mite resistant and conducive to the area, I only had to treat 2 hives out of 11 all season this past year. It is possible, but takes time, hoping none will need treatment eventually.
Yeah, so I didn't mention that my all natural treatment-free panelists, like my stakeholder group, really were sad that my research was short, right? So 2 years, they definitely knew I wouldn't find what I wanted to, and they were hoping that I would follow those lines for 5 years. So even though the project is ending this spring, I'm going to take all the treatment-free colonies and keep maintaining them, because what they're saying is that, you have high losses initially and then they get better and better at overwintering. So I would like to see that myself. And I also, Kate has her hand up, so I just wanted to see what she wanted to say.
You're muted, Kate.
Thanks.
Sorry, Kate, you're
Play by
muted.
the rules here. But there are studies out there that talk about pollen diversity as being extremely important in immunity. There was a study done by the University of Illinois that shows that honey can affect the gene expression in worker bees, in particular genes that aid in the defense against pathogens and pesticides. The P450 genes that are responsible for neutralizing foreign substances and stuff. So I think just wanted to throw that out there. There are a lot of studies out there that support studies on nutrition. And what else was I going to say? Anyhow, I don't remember what I was going to.
Well, let's see, we have a few minutes before our next speaker. I'm glad we were able to extend the time. A great set of talks. I'm telling you, the Penn State just does a wonderful job. I have been able to, well, we first saw you I think it was your summer series, last summer. And we look forward to staying in touch with your research as well as your outreach. And once again, Margarita and Robyn and John and Kate and Craig, we really appreciate your time and perspectives. It's clear from the comments that you guys have really sparked a lot of questions. Our local beekeepers have each maybe generated some new requests for help and advice as mentors, because we have people asking questions about starting hives, working on hives at higher altitude. And so you're going to be hearing more from some of our attendees as a result of this meeting. So Margarita and Robyn, do you guys have anything you want to close with before we say farewell?
I do want to have, I want to mention that we are advertised that we're going to organize another webinar series this summer. And the theme is going to be beekeeping around the world. So we are going to invite people from Brazil who, do beekeeping management of Africanized bees, from South Africa, from Kenya, from China. And we're going to have the opportunity to, learn from them how they do their, the management and, and talk to them. So I hope that, some of you can join us for that.
That sounds terrific. Robyn?
Just thanks for having us. We're learning from you since we don't know about New Mexico beekeeping very well. So in my dreams, I would love to visit and see some of your, your top bar hives successfully overwintering. We can't really do that in Pennsylvania, it's too cold. And everything that you experience. So it'd be really fun to visit.
That's great.